Was from Rutgers University. So Chris said that he wanted to have the previous title, and now for something completely different, but Ken beat him to it. So it's and just like Ken, it's quite a surprise. I mean, Chris Rose is known to us as somebody who works in wireless and communication systems. I mean, he's a fellow of IEEE cited for contributions to wireless communication systems theory. And a beautiful thing about the information theory community and communication community is they saw that they had to diversify or become extinct. And found all these beautiful problems in biology, for example.
Now you tell us.
Yeah, it's true though. I mean, it's really. I couldn't possibly try to read Chris's bio. It's too much poetry for me. But right now he's a visiting professor at Brown University, and he was at 18 Bell Laboratories for some time in network systems research. But I have to say, at Bell Labs, it says here that he regaled his peers with levitated center conductor HITC.
ITC, remember that? Remember that old people?
Okay. So superconducting cables, here's a day about something different. So.
All right. So thank you. What Sean didn't say is that I'm the crazy guy, but I now have a friend. You know, I didn't realize how much of a friend, and I didn't. Organizers, you did a really great job. First off, it's hard to follow, Ken. That's one thing. Second thing is that he set up some things. We didn't coordinate beforehand, but he set up some things that I want to talk about almost directly. So I want to give you an idea before we launch into it, and I won't say there will be no epsilons. I'm just not even going to go there and say that there will be no epsilons. But just to give you an idea of the sorts of things that I like to work on, that's a paper in Nature. People like me, meaning communication theorists, mathematicians, yes, but communication theorists, we generally don't publish in Nature. So this was about a problem of delivering information over distance. And that's what wireless is, right? Wireless is delivering information over distance. And this particular work, which you'll see the title, it was the title paper of this issue, Dear E.T., How to Communicate with Distant Aliens. The punchline is that it's really efficient to write things down in some sort of material form and hand it to folks from an energy perspective. And it's startlingly so. So that's the sort of thing, again, I'm in a wireless lab. We work on MIMO, Wi-Fi, all sorts of good things like that, but ended up here. So this is, what I'm going to talk about today is the beginnings of a sally forth in that direction. Maybe we won't ask one of the big questions at the end of it, but it's food for thought, and this is a perfect venue, I think, for this sort of work. Or, excuse me, we'll find out. If I'm covered in blood and tomatoes at the end, then it wasn't a perfect venue. All right, so molecular communication. Ken alluded to this, and I kind of poked him in that direction at the end there. So I got involved in this by looking at Drosophila, which is a fly, and C. elegans, which is a worm. And these things develop from a single cell, and they just magically turn into a full-grown organism with, I love the way Ken said it, with feet and head. Well, maybe not feet for Drosophila. Well, no, Drosophila has feet, but the worm doesn't. So I got interested in it and started reading a little bit, and what I came across was this paper by another Nobelist, I can't remember, Nusslein Pollard, and I think it's Christina Nusslein Pollard. And what she discovered, and I think it was in Drosophila, and I don't think elegans had anything to do with it, but she discovered these things called morphogenic gradients. What the heck is that? So it's just chemicals. You know how biologists are, luminase and vociferase. We put them together and they form light. Doesn't tell you squat. I just named two things that I slapped together and doesn't tell you anything about the mechanism. But it was a little bit more than that. And the basic idea is this. You got an egg. It's tagged on one side chemically. It begins to divide, and then at about the 16 cell level, things magically begin to emit these things called morphogens. So when you're close to the head, you know I'm supposed to be close to the head, and I follow a particular program. Well, I'm far away from the head. I need to follow another sort of program. So, you know, for folks like us, that's great. It's kind of like the cartoon, but it doesn't really tell us anything. And the first thing that popped up is, well, how are they talking? I mean, this is diffusion, and this is a very robust process. I mean, if I put the egg in a flow field and blow past it, does that change the development? No. So what are the physics of what's going on there? Now, one of the problems with being us, though, is that, and this is where biologists have it over, folks in this particular area, biologists can keep a number of things in their head, completely conflicting things, without exploding. Okay? I am definitely an F equals MA sort of guy. Give me a couple of rules, you know, and I'll elaborate it. So the problem, you know, if you want to look at it that way, is that I just couldn't get past the notion that one cell was talking to another. And now the question was, and if it was morphogenic gradients, meaning you're generating these proteins that, or signaling agents that other cells recognize, well, that's a channel, right? So, just like a physicist, right down to a problem, how do we actually talk about this channel? So that's what this talk is kind of about. But then, I'm not really going to do biology. I'm really thinking about potential applications, for this crowd at least. So, just so we know, bacteria and us, we are eukaryotes, prokaryote systems, they communicate. Bonnie, who you probably know, Bonnie Bassler, you know, she was one of the first ones. Quorum sensing, everybody knows what that is. Bunch of bacteria, they hang out, strep. Hangs out, yeah, we're strep. We're dividing, but we're kind of under the radar. And then at some point, we get above the radar and become virulent and kill things. They're talking to each other, all right? Bacterial films, you know, actually we're going to go to one of these ponds out here, right? So let's find some pond scum, right? It's a bacterial colony, meaning it's differentiated itself. Somehow it knows how to differentiate itself. So these things talk to each other, is the point. So there's usually by emission reception of some sort of special molecule. These can be very, very simple, like calcium ions. Or it can be a little bit more complex, like epinephrine. It can be even more interesting, like mRNA or DNA, if you want to think generationally. There are these things called glycans. Actually, I don't want to talk about glycans. They frighten me. And I'll tell you why later if you want to know. In any case, there's all these different sorts of things that can be used to communicate. And evolution has had, you know, a few billion years to play with this stuff. So energy is usually at a premium, so it tends to be efficient. So when you think about communication, maybe you want to, this is ubiquitous in biological systems, molecular communication. So maybe there's something to it. So maybe there's something we should analyze and take away and maybe use for ourselves. So there's and actually, you know, you get old and people suck you into being editors and steering committees and things like that. There's a burgeoning area. Anybody do know about I don't even know what it's called. Muriel probably knows what the journal is called because you're the editor-in-chief of the JSAC. But it's biological and blah, 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 something communications. But it has to do with molecular communications is the point. But it's about nanosystems. So, you know, you build things and things get shrink. Communication comes at a premium, comes at an energy premium. So how do you do the communication? So, you know, well, biology has done it already. So maybe there's something to be thought of there or looked at there, especially since in these nanosystems, the stuff that we're used to using, essentially the antenna sizes are mismatched. And you have antenna mismatch, you got energy inefficiencies and life is bad. So token exchange might be more efficient. I'm not banging on that particular drum. It's possible, but you know, we'll see. So the other thing is that who knows what will pop up. Maybe macro systems should be using molecular communication. So this is kind of motivational, kinda. But the basic notion for me, because again, resident second crazy guy, it might be intriguing science and it's certainly interesting engineering if it actually works. So that's the desiderata, the motivation for what I'd like to talk about. All right, so this is a brand new area, as I mentioned. And what that means is that there's a number of different people working in it, a number of different people coming at it from different angles. There's a number of folks that know zero about biology but hook up with a tame or not so tame biologist, or tame the biologists, because it's kind of hard to do. If you've worked with a biologist, you know exactly what I mean. And my problem is that there's so many, again, F equals ma. I want to understand one problem really well and then see if I can extrapolate that out or expand that out to other things. So what the area, my summary of the area is, you know, well, it's all of these things. Some people are working on networks already. Some people work specifically in quantum sensing but don't do the information theory. Some people work with, actually do the information theory before understanding what the fundamental substrate is actually doing. So it would be kind of like doing network information theory without understanding single channels, not having machinery for that. So that drives me a little bit nuts. And since, you know, I'm old and cranky at this point, all I want to know is there a unifying elephant, right? That's what I'm looking for, a unifying elephant. What is the fundamental, is there some sort of fundamental theory? So some people, when you talk about using these chemicals, we should measure concentration. That's the lingua franca of moving, of information transfer in systems, concentration of a molecule. Well, when you look down at the actual physical levels where these things are, the sizes, you're not talking about agglomerations of molecules, you may be talking about a handful. So you're not being intellectually honest if you say concentration as a continuous variable is what you're actually dealing with. So once you do that, you know, I want to do a fundamental framework, fundamental limits, and applications. So today it's going to be mostly about the framework, something about the fundamental limits, and then I'm going to kind of, you know, do that, you know, possible applications that might be interesting, because I don't know what they are yet. All right, so here's the basic organizing principle here. So energy use is fundamental. We can all agree on that, especially as communication theorists. Okay, energy without, if you have infinite energy, then, you know, we just go home, right? Infinite capacity. Where is energy in these systems? I'm going to say, and you know, this is reasonable, that it's in token construction, meaning the thing that you're actually, either you construct a particular signaling molecule like a protein, or you sequester something that's already out there in the environment and then release it. Either way, that's going to cost you some energy. There's transport. I'm going to, at least at first, only talk about passive transport like Brownian motion, but you could imagine transport. And the other thing is that this thing that I call inscribed matter, and that's what that Nature paper was about, the notion that you take something and you write information down in its structure, and it turns out you can do that with ridiculous efficiencies. So this is a unit. Muriel's seen it before, at least once, and people kind of laughed when I said, you know, bits per kilogram, but a piece of RNA desiccated, you can store about 10 to the 24 bits per kilogram. Now, you know, kilogram of RNA, that's kind of a frightening thought, right? But even a milligram is 10 to the 18th bits. So the point there is that this is an incredibly dense way to store information. So, you know, maybe there's something you can do with that. Maybe biology has already figured that out, that you can use this sort of dense information storage medium to transfer information in useful sorts of ways. Now, here's this other thing. So with this inscribed matter, things are written down, and there's an actual message embedded. Well, with that calcium, calcium, potassium, whatever sort of signaling agent, chloride, they're identical. But you can still transfer information with these identical molecules. So what's the thing that's left? The only thing that's left is, well, you can do concentration in a time-ordered sequences of concentrations. Well, once you go down that slope, you really got to talk about arrivals of individual elements. So that's the only intellectually honest thing to do, and kind of a, what is it we teach our probability students? Finest grain and exhaustive, right? That's the only way to really look at it. So, I mean, that's kind of premature, but it's never bad to hammer home one of the main points. Mean first passage time, the time it takes to get there, figures in heavily into the capacity of these channels. Okay, so what's going to happen today? I'm going to show you an abstraction. I'm going to show you some modeling, talk about energy a little bit, I'm going to really gloss over the bounds, but I'm going to show them to you. Some observations on some numerical results based on those bounds. We'll do some ballpark calculations. And here we go. So here's a diffusion cartoon. So Ken had a nice picture of a cell with a bunch of receptors around it over here. So just imagine that this is a cell, single cell, and it has a bunch of receptors. And these receptors are sensitive to, let's say, chloride, just for the sake of argument. These are individual molecules that are being released. I have them in different colors because some of them might be different. But the thing that I want you to see here is that some of them are identical, the same. And the notion here is we're going to ignore, we're going to do a real covert sort of analysis here. Rest in peace. One of the things that I remember falling in love with this book, especially with the network information theory portion of the book way back when, is that he drew a circle, drew a sphere around an object and said, well, you can bound the capacity, the incoming capacity, by the amount of power that's coming in and the noise floor that's sitting inside that receiver. So that's kind of what I'm doing here. We're looking at this target. Molecules are going to diffuse. They're going to be produced by something. Maybe it's a gland, maybe it's a cell. Don't really care at this point. It's going to produce something, and that stuff is going to be emitted, and it's going to drift, in this particular picture, over towards this region, and then it's going to be captured. Now, when it's captured, it's processed. I'm ignoring the processing. All I care about is when it's emitted, and what the payload is. And when it pierces this, what I'll call a capture boundary. So that's the arrival time. It's emitted at TK. So one of these previous ones that was emitted is received at SJ. Maybe the payload was corrupted, maybe not. I'm not going to worry about that too much. But that's the basic model. Emission time, payload, wander, 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 reception. Anything else you do by the data processing theorem, and I keep showing you, maybe you warned me that this is a broad audience. So data processing, everybody? Data processing, no. All right. So by data processing, anything I do after it pierces that boundary, and this is a memoryless drift process, is irrelevant. All right. So what you often see, however, is because diffusion by itself has kind of sucky properties, right? Like meaning the particle might not ever get there. Right? That's a bad thing. People introduce drift. So that's my fan there. In which case you almost always have finite mean passage time, and I'm kind of ignoring the whole universe out here. Let's just assume that it's along a pipe, and you're kind of flowing stuff along a pipe slowly, and it's diffusing as it goes. So once again, what we're going to concentrate on is emission, transport, and capture. And now I'm going to abstract it. I think that's the next slide. Oh, here. Let's see if this works. This is probably useless cartoonery for you guys, but my graduate student who has escaped to China did this. Come back. There we go. All right, so there's the cell, or whatever it is. It's emitting something. I put a boundary around this so that wherever it pierces, then we know what time it pierces at. And what I want you to think of is this is the code that you're sending. So you're sending something at this time, this time, this time, this time, this time, this time. That's a potential code word. And I could change around those times at which I emit things. These particular particles are completely identical. But what happens at the receiver is, because of the random nature of the random walk, redundant, is maybe this one takes longer to get there. This one gets there relatively quickly. This one flies across the medium and gets there first. So the point here is that you don't know what was released when, necessarily, right? So you'd say there's no information there, right? Well, not quite, and that's what we're going to show. But it is a harder problem, because you have identical particles, you don't know which one was released at the receiver, you don't know which one was released. Your code word is this sequence. Your received code word, or your received sequence, is this one. But there's no necessarily mapping. You can't recover the mapping a priori from that emitted sequence. So now you had the word definition. Now you have the cartoon. Let's get rid of the cartoon. Let's put the mouse where it's supposed to be. Anybody see the mouse? Mouse, where are you? All right, let's bring it over here. Now let's go back there. There's the mouse. And let's just bring you back. Goodbye. Don't hate me because I use Linux. All right, so what are we looking for? General multipurpose outer bound model, and we're going to use bounds to avoid a modeling morass. And that's one of the beauties of information theory, as you all already know, right? Mechanism blind. And that's one of the dangers when working with biologists, because it's like, again, it's like crawling through lint. There's a zillion different things. I hope there's no biologists in the audience. But it's really a lot of detail, and what I'm looking for is some sort of fundamental precept. Okay, so here we go. Here's our mathematical abstraction. There's our input. So that's the times at which we release these identical, for right now, molecules. Each one of these molecules is going to travel from sender to receiver with some IID delay. If we knew which individual molecule corresponded to which individual arrival corresponded to which individual departure, you'd have this. But that's not what we have. We have a sorting process where the actual output is the time-sorted version of these S1s through SMs. So just to throw the nomenclature at you, T's are departure times, S's are reception times. What you actually see are these unordered, or these ordered S's, S arrows. And we're going to say that there's a first passage time. The expected value of each one of these D's is 1 over mu, their IID, and I think I've said it all about the basic model. That's not too bad an assumption, as long as you don't have active transport. Okay, so now this is, what we're here for, right? Mutual information. Well, the first one that we talked about, S without the arrow on top of it, that's kind of easy, right? So you got M tokens on an interval, meaning T1, T2, T3, all the way up through Tm. And you just bust it up, right? So here's a vector S, vector T. It's equal to that, right? Information theory 00001. The conditional entropy of those, of the received, not the received, but the S based on T is just the entropy of D, the delay. Life is good. Oh man, how do we want to maximize that, right? We want to maximize mutual information. So, well, we want to make all of these guys independent. Don't. And that's been solved a number of times. The first time for this particular sort of problem that I saw it was Sergio and Venkat's paper, and I don't know, there's enough folks with at least a couple of gray hairs, and they're old enough to remember. Bits through Q's, that should ring a bell. So Sergio's and Venkat's paper, they dealt with this for a particular sort of model. In my particular case, I had to resolve it and ended up with some neat closed form expressions for this particular case. But the basic idea here is if you want to maximize the mutual information, you maximize the output entropy. Easy, right? I mean, we've done that. So that's done, and it's easy. For this particular problem, though, what do we care about? Well, what we actually care about is the ordered entropy. Now, the problem is not, you could start talking about ordered distributions. I hate ordered distributions. Okay, just do it. Just one of those intrinsic sort of things. Don't like them. But the real problem here, this one I'll show you is relatively easy, but this one is a pain in the bottom, right? Because if you look at this, let's look up here. Here, that H of D, those are all IID. The input has no control over them. None. Not so over here. Whether you can tell, the entropy of these ordered S's definitely has something to do with how you release them. If you release them all in a bundle, right, then, this thing, you have no idea which one corresponds to which. So that's the basic problem, and that's what has tripped up a lot of people before we decide to play.
And you have a sampled model, right? So they can arrive at the same time?
Yeah, this is continuous, so they could arrive at now, I take that back. Yes, they could arrive at the same time, right? But that's a zero measure.
Okay.
So I know where you were going, and I just kind of danced around to get to saying, no, there's no ordered model. There's no discrete model for this. And that actually helps us, and I'll tell you why in just a second. Thanks. That was just like we were anticipating some advanced stuff, you're anticipating mine. So here's what you do. You make, the fundamental issue is that, I guess that's kind of cryptic, but the bottom line is this. if you send all of these things out, right? There are m factorial ways of sending these t's out or thinking about how they're ordered. And I'm just defining a permutation operator here, and there's an index on that permutation operator, omega, going from 1 to m factorial, because you can arrange these things m factorial ways. All right, now here's the thing. Whether t, let's go back to, I'll say it and I'm going to go back to the picture, whether t was sent or some permutation of t was sent, you can't tell at the output. So let's go back to the picture, right? So let's say I just arbitrarily reordered these guys, right? So I have one set of t's that I sent out, and up on the next interval I'm going to send out the same set of t's, but they're reordered. At the receiver, you can't tell the difference because everything is ordered in terms of arrival time. It's not ordered in terms of place, is what I want you to think about there. So what that allows you to do is, well, if you can't tell them apart anyway, let's make sure that they're, not make sure, we can reorder or change the input distribution so that it's symmetric. And all that means, and I call it hypersymmetry because it's in higher dimensions, but it's just symmetric, that f of t, no matter how you permute the indices, is equal to the, so f of t is equal to f of its permutation for all possible rearrangements. And then what we're going to do is we're going to search. Whoops, here we go. So we're just going to search over only those hypersymmetric f of t's. Okay? All right, so that's the first insight. The second insight is, so the second insight is, well, if f of t is hypersymmetric, well, so is f of s. And yeah, it's not a big deal, it's a little proof. This is more important. It's easy, but it's more important. We're going to only assume the first passage times are non-singular, meaning a nice continuous distribution. Okay, we're not assuming that with some finite probability things get there at a particular time. So this is important. Once you do that, f of s is just a bunch of permuted patches, right? And there's m factorial of them. Now here's the punchline. Because fd is non-singular and fs is therefore continuous, the edges and corners of fs, they have zero measure. So now guess what? Fold, 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 fold. And we're done with h of s arrow. So h of s arrow, meaning the ordered version, is just equal to h of s, as if we knew which ones corresponded to which inputs, minus log m factorial. So we just knocked that away. But that's easy. That was the easy part. So what I'm first going to show you here, because I need to re-emphasize some nomenclature. So here's our channel again, our abstraction. There's omega. Omega is our reordering index, omega equal 1 to m factorial. And what we got here is you take the s's, you apply some permutation operator omega to it to get the ordered s's. That's the way to think about it. Well, you can think about this channel in operation as, well, that's a random variable, random vector. That's a random vector. And that's a random variable, discrete. All right. So here's something kind of interesting, and this is really the key and the nut of what's going on for all of what happens after. S, which is that set of things that we know exactly which S corresponds to which T, S1 corresponds to T1, S2 corresponds to T2, and so forth and so on. If I take S, then I can certainly generate the ordered S and know what the permutation operator is. And this bears directly on Muriel's question, can you have things arrive at the same time? That don't work if you can have things arrive at the same time. But that's a zero measure event, I'm ignoring it. Likewise, if you give somebody the ordered S and the permutation number index, you can certainly generate S. So those two things are exactly equivalent. What does that give you? Well, we just do the usual decompositions. There's h of s. Well, that's the same as h of this pair given t. Blah, 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 blah. What you end up with, there's this new thing that we defined. And what you end up with is the mutual information between the actual output that you care about, that you can actually see, the thing that you can actually observe, is equal to mutual information of the, did I say capacity? I meant to say mutual information, of the place of the system where you can see everything minus this stuff. Right? Or the way to think about it, as a communication theorist, we're going to try to maximize that beast. We know how to, this is the thing we want to maximize, and then we have these two things which are constant. So we're really interested in that. That's easy to do, especially since the D's are, the first passage times are IID. That's a little harder. And that's my student who escaped to China a number of years ago. He offered the first insight into how to play with that, and then we've expanded on that since. Now, there's some obvious tension in this expression. Not over there, because that's constant, but between that and that. And the tension is, entropy is maximized, and I'm being fast and loose, but entropy is maximized by independent T. That's what we'd like to do. Independent T1, T2, through Tn. The problem is that this guy is maximized by correlated T, and the simple sort of notion is, so that H of omega is the entropy, the uncertainty about ordering, given the inputs and the outputs. Well, if I launch everything at the same time, I have no idea which one came first. Okay? The ordering, we have no idea. All the orderings are equally likely. So that's not what I want to do if I want to maximize entropy. So it turns out if you actually try to do this optimization, in my hands at least, so optimizers out there, you know, help! But in my hands at least, I just can't do it. So I'm reduced to bounds. By the way, that was just, you know, that entropy is obviously log m when all of those things are equal. Okay, so I've had some struggles, right, over the years. First were a bunch of closed form results and bounds for that quantity, and mostly for exponential first passage, which is not Brownian motion, by the way. That, I have some lower bounds for the money shot in terms of calculating the mutual information. I have a really sucky upper bound, and it's better. It's sucky, but it's better than data processing. Data processing is just kind of irrelevant in this particular, wrong, imprecise. It doesn't help understand the basic problem. Obviously, it works. All right. So, oh damn, there it is. There it is. There's a song. What are you playing? So all I'm doing, this is for the information theorists out there, so that you know that I'm not lying to you right now, at least. So there's the model. You've seen it before, but here's the channel discipline. So what I'm assuming here, is that there's some epoch over which you send your tokens. Then there's some guard interval, and we're assuming that everything arrives by the end of the guard interval. Okay. And then we do it again. So it's multiple channel uses, and that guard interval makes it independent, right? Independent channel uses. And I'm doing like that, but what I really mean to say is that you can actually calculate this stuff out. One of the things that you end up needing to do, and this is where energy comes in, those tokens cost energy to make, so there's some parameter rho that's going to figure heavily, which is the number per epoch. So it's, you know, joules per sending interval. We're going to hold that fixed. And, you know, there's a bunch of limiting details that now I'm definitely not going to tell you, because there's epsilons all over the place. But here's the punchline, right? And frankly, I could have done all of that this way. Right? Because all I want you to know is that we're going to fix the number of things we launch into the channel per unit time, and that we must have the expected value of the mean first passage time be less than infinity. It's got to exist. Once you do that, life is perfect. Now, for those of you, if you were ever interested in this area or working in it, you say, but wait, what about Brownian motion? Sometimes it doesn't ever get there. Well, look, in a confined space, okay, wait for it, okay, you will eventually all get that cough, right? Because we're in an enclosed space. That's a lie because of the stupid vent. Stop it, vent. But the idea here is that the thing actually gets there. So we do some bunch of stuff. I'm sweeping a lot under the rug here, but there's some nice things that pop out of this. First off, we can define a capacity per token. So capacity per molecule. So, you know, I have to There's a bunch of papers. All conference papers. I'm just too lazy to write the journal papers. I guess I got to get off my duff. All right. Another neat result is that the capacity per unit time, nats per unit time, is just that how fast you launch tokens into the network times the capacity per token. Now that's, maybe that's obvious, but it's, you still got to make sure that it's true. Especially when, the bottom line is for asymptotic things. I'm speaking to the right audience. When you start taking m's and epsilons to 0 and m's to infinity, you got to make sure everything works. So it does. All right, so now let's do construction cost, and we're getting down to the real money here. So if you got identical tokens, I'm going to just name that C0 joules per token. And I'll talk a little bit about that a little bit later. Inscribed tokens, meaning I'm actually putting payload information into a token. Well, maybe there's some substrate cost to build the structure into which everything's going to fit. And I'm going to assume a linear cost with the number of bits. And the motivation there is, everybody knows how proteins are made? All right, well, DNA, rip, mRNA, copy, migrate, ribosome, stick it in one end, more or less. Ken's laughing, but it's a cartoon. tRNA, bringing over the little amino acids, and pop! Every three base pairs, every 3 bases, new amino acid, new amino acid, new amino acid. And that's a very well understood process. And it costs a certain amount of energy to add each one of those amino acids. So that's why I did that. Now, there's one other thing that might be of interest to you guys. If I'm actually putting a payload, like a packet, right, in a packet network, well, I need a sequence number, right? Otherwise, I don't know, or some sort of coding in there. Otherwise, I don't know which one corresponds to the first and which one corresponds to the last. So in packet networks, you just have some packet header field that's, tells you how many, some number. But with the asymptotics, where we're talking about using this channel m times, some number of times, and the size of the epoch over which you send m goes to infinity, well, log m, log m factorial goes to infinity, right? That's not good. Well, so you need sequence bits, but in order to work with all the previous machinery, here's what we know. This thing that we derived before for the identical token channel, that guy is a lower bound on the number of bits you need to send to recover the sequence of the packets. So we have the machinery to deal with that, is the point. So that's all this is meant to be. All right, so let's define a few things. This is kind of non-intuitive, but I'm defining it anyway. It's first passage rate. So 1 over that is first passage time. That's the rate at which you launch tokens in. We're going to call that chi. And I'm just going to give you a parade of things and show you a plot, or a couple of plots. One other thing, exponential first passage is assumed here, and the reason is that it's kind of like the Gaussian, it's kind of like the, I can't even say it, but you know what I want to say. Gaussians are bad for additive channels. Exponential is the worst thing, is the min-max for this sort of channel. All right, so one little motif for the math folk out there. So there's our epoch. That's how long our epoch is. We're assuming it's m over rho. We fixed rho, so the epoch goes as m over rho. This is just a little factoid for exponential first passage. That's the entropy. I actually had to correct that in Wikipedia. Somebody screwed it up.
That's great.
What?
Huh? No, it just...
There are errors in Wikipedia?
No. What? Oh my God. This, needed a little bit of derivation. Sergio has a similar formula, but he had a different model for the constraints on the input. And the end result is that the maximum mutual information, when you know, now this is for, I'm launching a token in here, and I know which one it is, and it comes out here at a particular time, the capacity of that channel is this. Okay, so I just want you to see, it's a loggy sort of thing, and it kind of makes sense. The way to think about it is there's some sort of finest grain resolution based on the first passage time. When you launch things closer than a first passage time, there begins to be some ambiguity. So that's the capacity of this simple channel. All right. So let's look at some things. I say remember this, and again, by this point you're completely almost asleep, right? This looks frightening, but it isn't. See that log chi? Well, go back here. See that chi? Go over there. See that log m factorial? What's that? That's m log m minus m. So you can see that the m in that previous expression kind of gets sucked away. Wave my hands, wave my hands, wave my hands. That is that h of omega thing calculated out for this particular case. Believe me, trust me. And that's a lower bound. And that was for timing only, right? So 1 over C0, that's the number of bits per token. Oh, come on, Rose. Yeah, that's kind of wrong, right? That should be CM. Sorry about that. As if you noticed. All right. All right, so payload only. What if you just sent payload only, like in a packet network, right? Just send the payload only. Well, blah, 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 blah. You end up with an expression like this. What if you now, in addition to payload, you use timing too? Okay. Well, same sort of expression. Now you have the timing capacity. That's your number of bits per token. Number of bits per token. That's the energy cost. And that's something that I'm going to plot. There's one interesting aside here, that if you label things with log m factorial, each packet, right? There could be m factorial ways it could arrive, so you label each one with a log m factorial over m piece of information. If you do that, the rate goes to 0. So there's something kind of sneaky and cool about the way we do packet sending in our actual networks, because there's a basic assumption that if I send a package today, it's probably not going to arrive next year, right? So there's kind of a basic assumption that's built into that way of looking at things, and that falls out of this math. All right, so here's almost the end of things. So what I plotted here is as a function of chi, so let's remember what chi is. As chi gets larger, it means you're sending fewer and fewer elements into this network per unit time. Okay, fewer and fewer elements. So it's a very diffuse, no pun intended, sort of communications. So what you see here are a bunch of plots. On the bottom here is payload, up on the top here is payload plus timing. B is the size of the payload in bits. On the bottom here is payload alone. And this one that doesn't change from plot to plot to plot is timing alone, as it shouldn't, because we're indexing in B. And the takeaway message here is that for timing alone, so if you just do payload, the info per unit energy, okay, bits per joule, is best when you can both send a little bit of payload and resolve which T corresponds to which S. Because that's what's happening here. We've sent enough information so that we essentially have a bunch of parallel channels, as opposed to this mishmash where we don't know what's happening at the output. All right, and it gets worse in terms of bits per unit energy as you make the payload larger and larger and larger.
Explain one more time. What's the dotted line again?
The dotted line is the, so there's a,
The other two.
I know, this is it's a really sucky plot.
Yeah.
But the bottom line, the dotted line is payload and timing. We're using both payload and timing information. So you get a real, it's not, so this number here is not just the, adding that and that. It's significantly larger because essentially we parallelize the system. Instead of mushy-mashing them, we now have independent parallel channels in this particular case. So that's the info per unit energy. And that's really the big picture that I want to take away here. And the more bits you add into your payload, the suckier you get in this model. All right, now let's look at rate per unit energy, which is something we might care about. So once again, this binary sort of, B equal 1, either on or off sort of thing, two different molecules is one way to look at it, does you well. Down here is timing alone. Right? So timing just, it looks like, analytically, it looks like it has a little hump, like there's an optimum right there, and then just flattens out. Here's if you're using payload alone, and the picture here is that for rate per unit energy, as you add more and more and more payload, the payload line begins to approach the payload plus timing line. Right? So that's kind of a useful thing. If you don't have to use timing and you put a big payload in there, just use the big payload. And what I like about this, despite the fact that I picked numbers out of the side of my neck, right, unit numbers, that's the cost. I'm assuming that the substrate cost is this, and the incremental cost is the same as the substrate cost. The cost function matters. But even with those simplistic sort of numbers, it's interesting. One other thing that I have to tell you here is that we're talking about, I flipped the abscissa, and now we're talking about how fast we're launching things into the network relative how fast they're being taken up. So this is more. So on average, there's maybe 10 things in the network at any given time, 10 things still in passage. And the capacities are interesting. These are actual. This is in nets. And now let's actually look at some numbers, because I've kind of given you no analytic detail here. I said, believe me. But now let me give you some numbers that you can believe me even less. All right, so we're going to play a little bit numerically. So here's the setup. I got a source. I'm releasing these things in a controlled fashion, these individual tokens. They're drifting down through a pipe. I made this one-dimensional. And they're being picked up at a sink. So T is when these things are released. S is when they're picked up. What I actually see at the sink, though, is S arrow, the ordered version of it, because I don't know which one corresponded to which just yet. Okay, so here's the number, protein construction. This is well known. You look at Leninger. Is an ATP is a unit of energy that biological systems use. So that's the amount of energy per token in this particular case. So 3.2 by 10 to the minus 19th joules. Right, so let that sink in. That's a small number. Now let's use some stupid stuff, regular old diffusion coefficient. So in air, where the mean time before collisions is a lot longer, things move, the diffusion constant is larger. In things like water, the diffusion constant is significantly less. I'm going to give you two simple examples. So I also need this mean first passage time is about that for this setup. There's things that I'm not telling you. All right, so if you want to send a molecule across a table, so I release it, and I send it, and I just let it go, and eventually it's going to go a meter away, and the room is completely still. There's no convection, no air currents, and we're all dead because that's what would happen. It takes about 14 hours. So this is, not really a great thing to do. You will need a fan for that particular model. Across a synapse, which is 20 nanometers, we're talking about faster, about 200 nanoseconds. All right, let's look at some capacity numbers. So for small payloads, B equal 1, and then I set the transmission rate equal to 1 as well. Across the table, using those numbers, you got bits per day at the attojoule level, right? So that's 10 to the minus 18th joules, bits per day. Not very exciting, right? I mean, bits per day. Well, maybe, maybe not. If you're talking about environmental sensing, maybe that's exactly what you need. But the picture there, the thing there is attojoule. Next thing, across the synapse. Well, now you got megabits per second per attojoule. Now, once again, I'm not talking about, for those who do neurophysiology, I'm not talking about upkeep of the individual nerve cells and repolarization of membranes, none of that. I'm just talking about constructing these molecules and putting them away. And frankly, in the neurophysiological context, it's not a protein. So I'm not even sure what it costs to, for the neuromuscular junction, pick up and develop a molecule of acetylcholine. All right, now let's make it a little bit bigger. We're going to send more molecules into the system. We're going to do 1,000 bits of payload. And now across that table, and again, that's tremendously pessimistic across the table, because really what I want to do is blow it across the table and decrease the mean first passage time. Now we're talking about kilobits per day per femtojoule. We're still way the hell down there in terms of energy. And across the synapse, we're talking about gigabits per second now per femtojoule. So, that's kind of interesting. And now we're going to go over to, and again, that's interesting, but again, I kind of pulled that out of the side of my neck. But where I think it really begins to get interesting is, let's look at some, remember that bits per kilogram number I threw at you a ways back? Let's look at some common substances, bits per kilogram. So 20-pound paper, if you put it through a 1,000 dot per inch printer, you can get about 10 to the 10th bits per kilogram. Yeah, it's easy to calculate, actually. A DVD, 10 to the 12th. Magnetic storage, just the film, not the disk, 10 to the 17th, getting up there. Optical lithography, 10 to the 18th. Once again, just the silicon dioxide, not its substrate. E-beam lithography, you can do quite a bit better, as you can imagine. If you want to laboriously take xenon and take xenon atoms and place them on a nickel substrate, you can get up there towards 10 to the 22. There's our old friend, RNA. And if you really are trying to get really cool and slick and just use lithium and beryllium, which are the two lightest elements, as if they form an alloy, you can get something really impressive. And there's always the old fallback, the black hole. And I actually had to, I didn't derive it. I ended up having it derived for me by, what's his face? Hawking. And but that's kind of useless and bad. It's not a very social way to communicate. Now, what this says to me, right, is that if you can tolerate a little bit of delay, I mean, here's how you should do your next Netflix distribution, right? And I guess, you can even wrap in, you can dance around and say maybe there's some fountain coding going on in there, right? And then some cool stuff about how do you recover it and recover the money. Basically, you send everything out as these molecules, and the information storage is so dense that you can send everybody, and this is, Terry, you said the way F-BIOS works, right? Send everything to everybody, and they just pulled it out when they need it. And if you need it there right away, right, you could do that. So that's the big
diffusion with drift.
Excuse me?
That's a diffusion with drift.
Yes, exactly.
Yeah.
So, you could do these variety of things, but now let's get a little bit, and this is where I think it could become interesting in a more controlled environment. It might be interesting in that other environment too, but let's think this way. Here's a disk farm, right? And I forget, I'm trying to think. No, again, so we're, so you and Ken together, we're as one, right? So there's a disk farm, and you know what all those things are, right? They're fans, cooling everything. What do fans do? They blow stuff. Right? Well, if they blow stuff, that mean first passage time, if you have a suitable substrate, goes way down. But again, we're not going to, maybe if the payload's large enough, we don't have to worry, because we're worried about bits per second. Well, 1 milligram of RNA per second, that's the data transfer rate. Dumping 1 milligram of RNA per second. 10 to the 18th bits per second. Right? So now the question is, the fundamental question is, how much does it actually cost to construct those sorts of messages? I don't know just yet. But when you're faced with numbers that are getting up there towards Avogadrian, I mean, this is many, 6 orders of magnitude off of Avogadro. But when you start getting up to those sorts of numbers, you got to begin to pay attention. And this should be your appropriately odd response. So what did I tell you? All of that stuff, timing plus payload. I've given you a framework, I've kind of semaphored a framework. There is a framework, but I haven't really told you the whole thing. Lower bounds. If you need bit efficiency, you should do slow release with timing or a small payload. If you need rate efficiency, you should have a large payload or just do fast release. And the whole point is that these rates are scary large and scary efficient. And when you have those sorts of numbers, regardless of whether you think this is stupid or practical or whatever, these sorts of numbers you can't just ignore, is the point. And that was actually the basis of that nature paper. We were talking about 10 to the 15th, 10 to the 16th, 10 to the 18th sort of efficiencies. All right. Now, I said that already, but I'm going to re-emphasize that. Transport efficiency. You got to worry about. Transport latency, right? But if you have a big enough fan, maybe it doesn't matter as much. So what's next? There's some technical annoyance about the upper bound. Excuse me?
Oh, no.
Yeah. Technical annoyance about the upper bound. Comparison, this is for all of those snake and the elephant and this. I just want to lay all that stuff to rest. So I haven't done that yet, but that's next. Now I want to really think about how this applies, or if it applies to biology. There may be places, because biological systems use individual tokens. They also use inscribed tokens. Why in one case and why not in another. So that's something that maybe this sort of analysis can answer. This is where I got to talk to a chemist, how fast and how much energy would it take to construct these really dense tokens. Ken was talking about it takes, about an hour for a doubling. So you figure, what is that, no, gigabases.
Yeah, but like, so cytokine secretion or something is that a phenomenon?
All right, so the point is it could be fast, and it's not like we use these cells as fusion reactors, right? So the energy is finite. And we paid no attention to any of that, and that's important. What actually happens, the kinetics of what happens at the receptor, and any information loss that occurs there. And then there's other people that are working on this, but I'd say that just to say it, and because biological systems are networks of multiple actors. For my part right now, unless somebody can drag me kicking and screaming, I don't want to worry about more than, I just want to do the myopic physicist, one channel sort of thing. All right, so here's one possible punchline. And this is also a punchline of, and everybody knows this, I think this was in Tannenbaum's book, for those of you who remember Tannenbaum's book before he fell from grace. Truck filled with storage media is a very high bitrate channel. And what this talk, I think, adds to that is that, a swarm of time gnats, with backpacks,
Oh, that's great.
in a breeze, right, is way cooler and could be actually way more efficient. All right, so thanks, and that's it. And not too far over. Let's go. Muriel,
So actually, when you were talking about the losses, presumably because some of the things you transmit might not make it to the receptor, right? Especially if there's some running around in that soup. So
haven't touched that.
do you think the deletion channel would be useful there?
Right. Yes, I think it would be useful. I haven't touched it because, again,
ugly.
well, it's not so much that maybe it is ugly, but again, I'm just, how it is.
Yeah.
It's just the anal retentive thing. I want to solve this problem. Right. So I haven't looked at that yet, but yes, a deletion channel model seems like it would be an obvious. Now, the worry there is, period. A deletion channel model is the obvious thing to do. I don't know how it would work, though. So, if you can tell me, that would be great. All right, Ken, and then Terry. Oh, Terry?
I'm going to ask you a few questions.
Uh-oh.
Put me in charge, Terry.
Can you go back to your slide where you had the fans and the servo rack? I didn't quite get what point you're trying to make there, or what
Okay,
how
so...
it applies to the fans.
So if you're trying to move information around in a server network, right? It's wires, right? So there's a couple of things going on here. One, the storage medium you're using may be not the best. You got this rotating machinery in there, right? So let's get rid of that. Let's make it all solid state sort of stuff. You still got to get information from one place to another, right? And that's wires, and that's heating, and whatever else. Well, maybe it's more efficient to first change the substrate. I mean, I'm changing multiple technologies on you right now. Change your storage substrate, and then instead of copying, just kind of blow it over. In other words, you have a surfeit of information. Milligram is nothing, right? So my point is that maybe with a surfeit of information, there are other architectures that one might be able to use. Did I answer your question?
Yeah, no, I get it now.
Oh, yeah, Ken.
Yeah.
Thank you, sir. I mean, the thing you pointed out right at the end is that you're only considering the one-to-one thing. I'm a bit confused about it because, like, in a biological system, it's not like there's two objects that know, I'm trying to communicate to you across this room, Chris. And when you're on the receiving end, it's all interchangeable, right? So pulling timing information, you're potentially,
All of the above. But the question there is, it's certainly the case that there's information going from one place to another, and there's a physics of information as opposed to a practice of information transfer.
Sure.
So I'd like to understand the physics first.
Okay. And so can I ask a second question?
No.
And even with the one-to-one thing, is that, if it was timing was the thing that you're doing,
If.
then the original cell or whatever making those things would have to have a good regulated way of changing the timing at which it secretes things. I was wondering, is there any evidence that actually the cell can do things?
No, if you look at, so for example, so I haven't done, now here's I shouldn't say no, because every time, and I'm sure you know this, every time I say no about a biological system, my tame biologist, Syra, comes up and says, yes. Right, but yeah, that's something, if you're talking about a regulated and timed way, neuromuscular junction, you already know, it spits it out. But that's the sort of thing, right, you might want to be interested in. Why do you collect these things in vesicles in that particular case, And that's definitely a high rate sort of thing, right? Diffusion, it's like lion. And diffusion doesn't really help you in terms of, so you need something that operates fast, lion, run. So the way I look at this work is it's going to inform the energetics, and then you look at the biology and say, in this case it uses this, in this case it uses that. Is there any correlate? Modulo everything you said, you got systems of things, right?
I'm just going to make one last point.
No.
Related
We're done. This is discussion, right?
relation to Muriel's question. So, what happens in the blood, so inside yourself when you signal, is in fact that signal is degraded super quickly because protein secreted into the blood is removed very quickly. If you produce too much protein into your blood, it crystallizes and will kill you. So you have this complicated system that kills it very quickly. So with lymphocytes, for example, they give inflammation signals, but they do it very locally. So like you get an infection, what happens is the initial proliferation happens in your lymph nodes,
Right.
but actually then they circulate in your blood, and it's only when they get close to the actual site of the infection that they expand. And the reason for that is there is signaling there. The signaling is very short distance.
Right.
I don't know bits and pieces about the nature of those distances, but I guess that's what I had in my mind when I was wondering about, losing individual tokens from a single cell seems very likely. And then also, I guess my final question to you is about, even if you have two objects communicating, you're kind of presuming that they need to have a very refined piece of information at the end of it, where the cell may not actually have a huge range of options available to it. It might just be deciding, should I expand or should I not? Or
No, right. No, completely agreed is the point. So this is kind of, like most of my work, this is slightly schizophrenic, right?
yeah, okay.
Right? So one thing is looking at the fundamental attributes of the channel, and then maybe we can lift that and play some engineering games on our own. But with the fundamental physics, could be that when we look at the biological systems, there's going to be specific information theoretic reasons, information theoretic physics energy reasons why the system operates the way it does. So that's not, I haven't even touched that because again, anal retention, myopia. So, and I make no apology for that yet.
Deliberate.
Yeah, deliberate, that's,
Thoughtful.
yeah, but we're among friends, it's exactly what it is.
Do you mean apology in the sense of defense? Anyway, no, sure, I wasn't asking you for apologies.
Another question? Well, yeah, thank you again.
Thank you.